Hello everyone. Today I'm going to discuss about the preparations for light microscope examinations. Just the basics of the and also some of the techniques such as gram staining, simple staining, and negative staining, which I'm going to discuss now. And I am Vivek Patni. Also, please subscribe this channel. Okay, so starting with the basic and the easiest one, which is known as wet mount. Okay, it is the simplest type of preparation. Okay, in which a specimen is placed on a slide in a drop of a liquid. Okay, a specimen is placed on the slide in the drop of a liquid. Okay, for example, take a slide, simply put a drop of water, any kind of water sample, it will be your specimen, or any urine, even blood, whatever it may be. Okay, solid specimens such as screens cappings can be also placed on the slide before adding the drop of the liquid to prepare the wet mount. Okay, uh, simply the liquid used is simply water, but often stains are added to enhance the contrast of the specimens. And the once liquid has been added to the slide, a cover slip is placed on the top and the specimen is ready for the examination under the microscope. So this is how a wet mount works. Okay, and there are some of the benefits. Okay, live and unstained cells can be seen under the microscope. Okay, and also we can also find out the behavior of the cells. Okay, because some of them are live ones also. Okay, but the problem is with the magnification. Okay, we cannot achieve a higher magnification in this case, wet mount method. Okay, so next comes other smear preparations, that is bacterial smears. Okay, and basically what is a smear? A thin layer of solution, a thin layer of a solution of microbes on a slide is called a smear. Okay, please check the photograph here. Okay, this is a smear. Okay, this here. Okay, the slide is here, and this one is a smear. A smear is usually fixed to attach to the microbes to the slide and kill the microbes. Okay, after it is very simple. Take a drop of your sample, or let's say cultural solution, okay, bacterial culture solution, or you can even use a water sample. If you are going to use distilled water, you will not able to make smear. Okay, so smear, basically take your sample, which is generally a liquid one. Okay, which is basically a liquid one. Okay, and simply spread it on the slide using the nichrome wire loop. Okay, using the nichrome wire loop, it is the best way to prepare a smear. Okay, and after that you need to fix it. You can fix it uh, by use of heat. Okay, most common method. Also use various chemical agents also. Okay, chemical agents such as acetic acid, ethanol, methanol, formaldehyde, glutaraldehyde can denature proteins and stop biochemical reactions and stabilize cell structures in the case of tissue samples. Okay, so this is how smear is prepared. Okay, just the basic introduction. Okay, and now next comes the types of staining. Okay, there are very different variety and different types of stains. Okay, negative stain, simple stain. Okay, we'll study it more detail now. Differential stains, of which two of them are gram stain, acid fast stain, and other special structures such as two-view capsule stain, endospore stain, and flagellar stain, which I'm going to discuss. Okay, before moving forward, if you are watching this video, please download the Science Edutech app. Please download the Science Edutech app for for if you want to study the basics of microbiology, if you want to study the basics of microbiology and purchase the course but basics of microbiology. Okay, and also subscribe my channel, okay, to view more updates regarding the other videos also as well, and also subscribe Sagar Sonara's channel. Okay, and the link for the Science Edutech app is given in the description itself. Please check out. Thank you.